Well, hello everyone, and welcome back to Science in the Age of COVID-19. We hope that everyone is staying safe, and we hope that our seminar series, in addition to providing knowledge, can also grant you a little bit of sanity in these crazy times. Today, we are super excited to welcome Scott Hensley from UPenn. Scott was, a theme emerges. Of course, no one used to work on SARS-CoV-2 until they did. Scott spent most of his career focusing on influenza, particularly the mechanisms of antigenic drift, one of those terms that no one used to know, but now everyone recognizes and worries about. As soon as the pandemic arose, Scott retooled his lab to focus a bunch on COVID. They've done a bunch of stuff working with convalescent plasma, looking into infections in maternity wards, deep immune profiling, the impact of seasonal coronaviruses. And I think that Scott is going to tell us a lot about the antigenic drift today, particularly the new variants that are keeping us all up at night. So Scott, thanks so much for coming.
All right, Loren, well, thank you for that introduction, and thank you for having me here today. So the title of my talk, Immune History and SARS-CoV-2 Antibody Responses, Lessons Learned from Influenza Viruses. And I know this is going to be a little bit unconventional, but what I thought I would do, since this is a COVID-19 seminar series, I would actually talk about flu, at least for the first 10 or so slides, because I think we have a lot to learn from other viral infections. And although SARS-CoV-2 is clearly a much different virus than some of the other viruses we've studied in the past, I think we can learn a lot from our prior studies. And today I'd like to show some flu work for the first, again, like 5 or 10 slides. So hang in there. And I'd like to try to draw some parallels to what might be going on with this new virus. All right. Okay, so I want to start today by introducing you all to Tommy Francis Jr. And many of you probably already know him. He wrote this famous essay published in 1960 on the doctrine of original antigenic sin. And Tommy Francis is one of my scientific heroes. You often get this question, if you can go back in time and have a beer with anyone, who would it be? For myself, it would probably be my 2 grandfathers who died when I was fairly young. But number 3 is probably Tommy Francis. This guy was a beast in the context of antiviral immunology. And on the doctrine of original antigenic sin, this was I think one of the most elegant findings as it relates to flu. And what he found was that when humans are exposed to antigenically distinct influenza viruses later in their life, that they have recalled responses, they have boosted antibody responses against influenza strains that they had seen earlier. It's pretty simple, but this is something, this idea of original antigenic sin, as you'll see in the next few slides, have been regarded as one of the most important findings, again, in as it relates to flu biology. Tommy Francis. Everything I'll show you today is consistent with what this guy thought of 60 years ago. Okay, so we are all exposed to influenza viruses by the time we are 5 years of age. This is a Sero-prevalence study from the Netherlands, conducted in 2011. And this is a country where childhood vaccination, at least back then, was rare. So we have H1 viruses and H3 viruses. They circulate in humans. And you can see that some of us are born with antibodies against these viruses. We can thank mom for these. These are maternal antibodies. But maternal antibodies don't last forever. You can see they decline over time. But again, look at this. By the time we get 4 or 5, now we have antibodies again. And that's because we're all exposed naturally to flu viruses when we're little kids. So what happens during these initial influenza infections? So here is a schematic of number of B cells in this case and on the y-axis, and time since antigen exposure on the x-axis. So upon influenza exposure, what happens? Well, the beauty of our adaptive immune system is that we have zillions of B cells, different types of B cells, and zillions of T cells. And we only have a few that are specific to each pathogen. And the beautiful thing is that we can recognize any pathogen that's thrown at us. But here's something that's remarkable, that upon antigen exposure, so here a flu exposure, these naive B cells, they expand like mad, and then they contract into what we call this memory B cell pool. Now, there's two notable things here. Number one, this memory B cell pool, there's more of these guys compared to prior to exposure. That's important. But what I would argue is even more important is that these memory B cells have such a low threshold for activation. You just tickle these guys a little bit and then they explode. In the case of influenza viruses, these memory B cells that are laid down from our initial childhood infections, they dominate all of our responses to flu later in life. You could be a 42-year-old person like myself and exposed to a virus in 2021, and guess what? Memory B cells that were laid down in the late 70s, early 80s in my body will respond to these antigenically distinct viruses that I encounter today. So memory B cells, again, there's more of them, but I think this low threshold of activation, how they can just take off and dominate a response, is something very important that most of us don't think about. So this is a nice review that was written by Matt Miller, and I like to show this because I think it's a nice schematic that shows the observation of original antigenic sin. Here you can see an individual experiences an influenza virus and then a drifted virus, so that's called virus A prime, and then is infected again with another drifted virus. So if we look schematically, what happens to that person's antibody response over time, you can see virus A, it causes an infection. You get this antibody response shown in dark purple. But now if that individual is infected with the drifted form, what happens? Well, you get a de novo response shown here in this light purple, but you can see a boost in antibody responses to that first strain that individual encountered. Now, during a tertiary infection, what happens? You get de novo antibody responses, but now you're boosting antibodies against that second infection, and you're also boosting antibodies against that first infection. There's always highest antibody titers to that virus that individual saw first in life. Okay, and there's, again, something special about these childhood infections. We don't understand it. Why is an infection that occurs when you're 5 years of age, why is that more important in shaping your immune memory compared to an infection that you encounter at 10 years of age? These are unanswered questions in the field of influenza immunology. Okay, so here's something remarkable. We actually have a pretty good idea about what types of influenza viruses have circulated over time. And if we look over the last 100 years, this is the timeline. Okay, so 1918, that was a pandemic caused by an H1N1 virus that circulated for 40 some years. How was it able to circulate? It was able to circulate because that virus continued to acquire substitutions in the outer proteins, in the case of flu, hemagglutinin and neuraminidase, so that could reinfect the host over and over. That virus was replaced with H2N2, circulated for a short amount of time. H3N2 came onto the scene in 1968 and has stuck with us since 1968 over the last 50 years by, again, changing in the hemagglutinin and neuraminidase proteins. You can see since 1977, we've had co-circulation of both H3 and H1. Now, I told you, I tried to really set this up that our initial infections are so important for shaping the types of memory that we have to dive into throughout the rest of our life. And guess what? If you were born in the 1920s, it is almost 100% chance that your immune system first was introduced to influenza viruses in the form of H1N1 virus. And if you were born in the late 60s, it's almost 100% chance that your body was introduced to H3N2 viruses as the first exposure to flu. And what we know in the case of flu is that these initial infections, they establish these lifelong memory responses, and they're very important for shaping how we respond to antigenically distinct strains and our susceptibility to pandemic influenza strains. If there's a new pandemic, we know that individuals that are exposed to these blue viruses, these H1 viruses, for example, are going to have different susceptibility compared to individuals that were exposed to these red viruses. So I just want to show you just a few more slides on flu, and then we'll get into SARS-CoV-2. But I just want to show you a schematic about how prior exposure can affect the susceptibility to our last flu pandemic, which was the 2009 pandemic. Okay, so in the case of flu, our favorite animal model are ferrets. And if you expose a ferret to the 2009 pandemic H1N1 strain, pretend there's just 3 epitopes that can be targeted by the human immune system, or I guess the ferret immune system. You have this circle, you have this green epitope, you have this blue epitope. Now, if you expose a ferret, you do have an immunodominant antibody response. It tends to target these highly accessible epitopes on top of the hemagglutinin or the HA protein, this red epitope. But you take my first grad student, this is work that we did when I started my lab 10 years ago. 10 years ago, if you take my grad student and he gets exposed to that same virus, he doesn't mount red antibodies. Instead, he mounts these antibodies against this green epitope. And if you take someone older, Tony Fauci, and I feel okay showing this slide today because I showed this slide at the NIH at a seminar where Tony was at several years back, and he had a laugh at it. But look, Tony Fauci is not the same age as Yang Li, and you expose Tony Fauci to this virus. He doesn't have red antibodies. He doesn't have green antibodies. Instead, he has blue antibodies. What the heck is going on? Well, hopefully your hypothesis at this point, after hearing me ramble for the last 10 minutes, is that this has to do with immune history. Well, it sure does, because you take ferrets, and guess what? If anyone that does animal experiments, you have a heart attack if you hear that there's an outbreak in your animal facility. We only use clean animals that are pre-screened and have never been exposed to virus before. And during primary infections, you do have an immunodominance towards the top of viral glycoproteins. It's interesting. We don't understand that. But Yang Li, he was born in the 80s, so his immune system was first exposed to influenza in the context of a virus that looked like this. See this epitope here? This is black, this is red. This is black, this is blue. The only conserved epitope is this green epitope. And if you are primed with this 1983 virus and then you are boosted essentially in 2009, years and years later, what we've learned is that you have a recall of B cells that produce antibodies that recognize those rare cross-reactive antibodies, those rare cross-reactive epitopes between these two viruses. So you essentially have an immune focusing on the epitopes that are conserved between these pathogens. What about Tony Fauci? He wasn't born in the 80s, even though he looks like it, right? This guy is amazing. He acts like he's 30 years old, Tony Fauci does. But he was running around Brooklyn in the 50s, and he was exposed to a virus like this. And you can see this epitope is black. This is red. This is black. This is green. The only conserved epitope here is the blue epitope. Same thing, right? He has this memory B cell response. Now, 50 years later, he gets exposed to this virus, and he has this immune focusing on these epitopes that are conserved with what he saw as a child. So we're all walking around with this immunological baggage. You could say it's baggage. It's actually a good thing to have these responses. The problem, the real original antigenic sin, you know what that sin was? Tommy Francis made it clear that sin wasn't that you were exposed early in childhood. The sin was that you were only exposed to one virus. And that wouldn't it be a dream to make vaccines where you can train your immune system in a way that you can have broad immunity to many different viruses? We're working on that in the context of flu. All right. Those that came for the COVID-19 talk. Here you have it. So I've tried to set this up that immune history, particularly immunity elicited by childhood infections, influence the specificity of your influenza antibody response. Could something similar be going on with coronavirus? And so as we start to think about how age and immune history might impact immune responses to SARS-CoV-2, I think there's 3 striking things about SARS-CoV-2 that could at least lead us to believe that there might be something to this. Number one, there's heterogeneity in disease outcome after SARS-CoV-2 infection. It's remarkable, right? Like some people are just asymptomatically infected and other people die. There's heterogeneity in the immune response after SARS-CoV-2 infection. Our group showed this in these 2 publications. These are studies that were led by Nola Meyer, John Weary, and Mike Betts, and we played a small part characterizing antibodies in these individuals, but other people have shown this as well, right? If you look at severely ill individuals, there tends to be higher antibody titers compared to people that are moderately infected with SARS-CoV-2. But look at the heterogeneity. You have some people that have severe disease that have screaming high titer. These are all people in the ICU for COVID-19 infection. Some people with screaming high titers and other people that don't mount an antibody response at all. That is a hallmark of SARS-CoV-2 infection, this heterogeneity in the immune response. And finally, there's this age-related or perhaps year of birth related, it's too early to really know, and those are actually different things, differences in mortality. And this has been widely described, and as shown here in this publication from last year, that older folks tend to have higher fatality rates compared to younger folks. Now SARS-CoV, I think everyone knows this, but SARS-CoV-2 is a beta coronavirus, and it's not the only coronavirus that has circulated in humans. In fact, there are both alpha coronaviruses and beta coronaviruses that have circulated in humans for years. So as far as what is commonly found, we have these two alpha coronaviruses, NL63 and 229E. And as far as beta coronaviruses, OC43 and HKU1 are the ones that have been widespread. And we all know about the original SARS-CoV and MERS, which have circulated on a much more limited basis. Now, how accurate are these estimates when we see, the emergence, when these viruses actually emerge? We really don't know as much about coronaviruses as we do of flu. So how accurate are these estimates? And I think we're going to learn more about that as time goes by. And second, how much do coronaviruses drift over time? Because I just told you, Tony Fauci and my grad student Yang Li have different types of antibody responses just because they were born, 30 years apart. So does it matter if you were first exposed to coronaviruses with the 1980s OC43 infection? Does that change if you were exposed to a 1995 version of OC43? I'm not going to answer all these questions today. Today is going to be a super speculative seminar series which is in line with, I think, the goals of the series. But I think these are really important things to think about and things that we are trying to address in my lab right now. But I won't have all the answers today. Okay, so some studies do suggest that recent coronavirus infections may be associated with protection from SARS-CoV-2. And this is a very nice study in JCI that compared electronic health records. And what they found was that individuals that had documented recent infections with these coronaviruses that I just showed you in the previous slide, that they were associated with reduced disease severity upon SARS-CoV-2 infection. Okay, so we know, to summarize, we know a lot about flu, how prior exposures, particularly those in childhood, affect our immune response. We know in the case of coronaviruses that there are different coronaviruses that have circulated in humans, and at least there's some circumstantial evidence, as shown in this paper, that some of these exposures, and particularly maybe in coronaviruses, the more recent exposures are associated with protection. So we wanted to dive into this in more detail. So the first thing we did was we measured antibodies to alpha coronaviruses and beta coronavirus spike proteins. And in this case, we tested antibodies from relatively healthy individuals that were collected in 2017, so before the COVID-19 pandemic. And you can see each circle here is a serum sample from an individual. We tested samples from a wide range of different age individuals. Consistent with what we see in flu, most of us are exposed to coronaviruses by the time we're 3, 4, 5 years of age. You could see these samples were collected in 2017. So, by the time you could see year of birth, 2010 or so, maybe a little bit later for 229E, most of us have antibodies against these viruses. Now, there's not a huge age-related effect here other than seroconversion occurs during the first few years of life. You can see that older folks tend to have approximately the same level of antibodies against these coronaviruses compared to younger folks. So we took these same serum samples, again, these were collected in 2017, and we completed similar assays, ELISA-based assays, using proteins from SARS-CoV-2. So we tested antibody reactivity to the full-length spike protein, to the spike receptor binding domain, and against the N protein. And what you can see is that about 4% of us were walking around before this pandemic with cross-reactive antibodies that could react to the full-length spike protein of SARS-CoV-2. Fewer of us had antibodies that could react to the receptor binding domain. And in fact, as I'll show you in a bit, most of these antibodies are directed against the S2 part of the spike protein. And a pretty large fraction of us, 16% of us had antibodies against the N protein. And the N protein of SARS-CoV-2 is more conserved to other beta coronaviruses compared to the conservation in the spike protein. So that is not totally unexpected. But in general, these data show that there's a pretty high level, 20% in all, of cross-reactive antibodies. 20% of us had cross-reactive antibodies before the pandemic. Now we've stratified this by year of birth, and there doesn't seem to be a year of birth effect in levels of pre-pandemic antibodies. So you can see here's the full-length spike protein, the receptor binding domain protein, and the N protein. And you can see there's pretty good distribution across all different age groups. Might seem like there's more circles here in the younger folks. Well, that's just because this was the population that we looked at here. We looked at a lot of kids. And so the line here is the percentage. And you can see it dances around a bit, but it is pretty much low levels across the board, across different age groups. Now, what do these antibodies do? I'll show you in a slide or two that most of them target the S2 domain of SARS-CoV-2 away from the neutralizing epitopes in S1. These antibodies are non-neutralizing. Okay, they bind, but they do not neutralize virus infectivity. Here's our experiments completed by Paul Bates. He has this VSV pseudotype assay where he is measuring neutralization activity of people that have survived SARS-CoV-2 PCR-confirmed infection, as well as antibodies in these pre-pandemic samples. And you can see there are some, these neutralization assays aren't perfect. There's some noise at these lower levels, but there really no appreciable level of cross-reactive neutralizing antibodies in these pre-pandemic samples, and there is certainly not any higher in this cross-reactive group compared to this non-cross-reactive group. Now we have, these assays are a bit noisy. We have completed BSL-3 level assays, and we see the same exact thing, that individuals that have survived SARS-CoV-2 infection have neutralizing antibodies, but these pre-pandemic samples do not have neutralizing antibodies. Those BSL-3 level assays were completed by Sarah Cherry. So this is a little bit different than what's been published. It's a lot different, I guess. What has been published in a recent Science paper with samples from the UK. And in that study, they found higher levels of cross-reactive antibodies in kids, and they found that they were neutralizing. And those two things we don't see at all. We tested hundreds of kids in this study. Not sure what's going on there, but where there's further investigation, because I think it's important as we think about these antibodies and protection. Well, this is what happens in vitro. What happens in vivo? And this is where our study gets super interesting. How would you ever design an experiment to see if pre-pandemic antibodies were associated with protection in vivo? You would do this. And we were lucky enough just to have access to these samples. And so the Penn Medicine Biobank, their slogan is, "A few minutes of your time and a simple blood draw can impact the future of medicine." And we've been working with the biobank for a while. It's a fantastic resource. I like to joke around that if you go into the hospital of the University of Pennsylvania just to use their restroom, chances are someone's going to try to run up to you and consent you to donate blood. And that's an exaggeration, but there are people that are just coming in for routine surgeries, people that are very sick. All walks of life are getting consented for blood draws constantly at our hospitals at Penn, and these samples are banked, and it's an amazing resource because they're linked to electronic health record information. And the consent form is nice because it allows us to reconsent those individuals. And so what did we do? Well, there's thousands and thousands of these samples banked. We identified pre-pandemic serum samples from 251 individuals who went on to become SARS-CoV-2 PCR confirmed infected during that first wave of SARS-CoV-2 activity in Philadelphia. 251 of those. And then we chose match samples from 251 individuals who did not become infected. age match, disease match, whatever. This includes samples from 80 COVID-19 individuals hospitalized with different degrees of disease, so we can look at disease severity as well. It's, again, just if you were to draw up an experiment where you would want to test whether or not, a correlative protection type of study, which this is what you would draw up. Of course, when these samples were being collected, we had no idea that we were about to have a pandemic. Okay, so when we looked at the entire cohort, this is shown here in the open circles are cases. So these people had COVID-19. In the closed circles, you can see these are the controls. There's no difference. When we look at antibodies against the receptor binding domain, you can see this is a very low frequency type of thing, as I showed you in the earlier slides. When we look at the spike, full-length spike, again, no difference in titers between the two groups, no difference in N protein titers, no difference in OC43 spike protein titers as well. Now, these samples were collected over several years, and studies have shown that antibody responses against coronaviruses can decline over time. And so we looked at part of this population, only those that had donated blood within a year before the pandemic. So looking at samples, only the samples that were collected more recently. And again, no differences at all, right? And there are similar levels of cross-reactive antibodies in both groups. And further, we have gone on, and I'm not showing this here today, but you can see it in this manuscript and in the supplemental tables. We've looked at disease severity, so hospitalization, for example, need for oxygen. Again, no differences in pre-pandemic levels of antibodies in either of these groups. So it seems like a lot of us have these antibodies, but they're not associated with protection, okay? We're just as likely to get infected and have severe disease upon infection. So what happens to our antibodies then upon infection? That's what I want to address next. And again, I was part of this group at Penn that published this study earlier, in September of 2020. And this included samples from 125 individuals that were hospitalized for COVID-19. And so here I'll show you some new data where we completed in-depth antigenic analysis with samples from some of these patients. And so these people were hospitalized, again, with SARS-CoV-2 infection. And so we tested serum antibodies at day 0 of hospitalization and day 7 of hospitalization. You can see when we do ELISAs with 229E, this is an alpha coronavirus. There's no change in titer over that course of hospitalization, no change in NL63. But look at this, OC43 titers. Across the board, we have rises in OC43 titers. And of course, we have rises in SARS-CoV-2 titers as well. These people are infected with SARS-CoV-2. So you see this concurrent rise in SARS-CoV-2 antibodies and OC43 antibodies, suggesting that SARS-CoV-2 is boosting an antibody response against OC43. These are the same data, just expressed as fold change. And again, you can see that there's a large rise in OC43 titers over the course of hospitalization. Now, I haven't introduced you structurally to the spike protein yet of SARS-CoV-2. Well, here it is. And as I've indicated, there's, broadly, you can think of an S1 domain being up here where the receptor binding domain is. This is the region of the glycoprotein that attaches to the cellular receptor ACE2. Most neutralizing antibodies target this region and sterically prevent binding to ACE2. The S2 domain is lower here. This is where the virus membrane would be, right? So it's more proximal to the virus membrane. And antibodies against S2 are not as neutralizing. And so we wanted to see which types of antibodies were being boosted. I told you OC43 antibodies were being boosted. And what we found by in these experiments that I'll walk you through is that these antibodies target S2, which have some homologous epitopes when comparing against these previous beta coronaviruses, OC43 and SARS-CoV-2. Okay, so the same cohort I just told you about, day 0 and day 7 of hospitalization. These are OC43 proteins here. So we tested the full-length proteins. These are the same data I showed you as on that previous slide. You can see you get a boost in the titers. But now, when we created recombinant proteins that only express this S1 part or the S2 part, you can see the S1, there's no boost in titer, but but that boost is all coming from the S2 domain. And again, the ratios or the fold change is are shown here. Okay, so we have these pre-existing antibodies. In case of spike, they're against the S2 domain. Don't seem to be associated with protection. And upon infection, we see these boosts. What about disease severity? No difference. Okay, so those that went on to survive out of that cohort I just told you, you could see here are the absolute titers. This is OC43 antibodies. You can see that there's really no difference between those that either the starting titer or the ending titer and those that survived versus died. And this could go either way, really. In the case of influenza, there's a lot of focus on antibodies against the HA stalk, which is analogous to the S2. And, most of those antibodies don't neutralize either. And I get worried to have, an antibody response totally skewed towards non-neutralizing non-neutralizing epitopes. And that's because, antibodies like that, they often work through processes like ADCC. You get a lot of tissue damage. You get a big cytokine response in those kind of scenarios. So this, in my mind, this could have gone either way. OC43 reactive antibodies against S2 and S2 skewed response, could that either lead to more disease or less disease? In this case, we really don't see much of a phenotype one way or the other. Okay, so to summarize so far, so humans are infected with seasonal coronaviruses just like they are with flu by the time they're 5 years of age. 20% of us are walking around before the pandemic with cross-reactive antibodies. There's no obvious age-related difference in these antibody levels. But here's the most important part of what I've showed you, and it goes against some published data, that these pre-pandemic antibodies do not neutralize virus in vitro, and they're not associated with protection. And they're there, and then upon SARS-CoV-2 infection, you get this huge boost of antibodies that react to the S2 domain, which is consistent with Tommy Francis's doctrine of original antigenic sin. These boosted S2 antibodies are not associated with disease outcome. Is this the end of the story? Of course it's not. So further studies need to be completed with memory B cells and T cells. And I think that is the key here, that these memory cellular compartments are probably what is involved in some of the heterogeneous differences we see in susceptibility and disease outcome. Because look, you want to go back to flu. In 2009, we had this flu pandemic. If we would take samples pre-2009 and complete neutralization assays, humans didn't have many neutralizing antibodies against the 2009 pandemic H1N1. Yet there was a boost in neutralizing antibodies in many individuals upon 2009 infection that recognized conserved epitopes with older seasonal H1N1 strains. So you in that case, you couldn't find that cross-reactive antibody in the serum component because it was probably such a low frequency antibody, but it certainly was in the B cell component, and that expanded upon that infection with the pandemic H1N1. Okay, so I'm gonna shift gears, and I see we have about 10 or 15 minutes, and I'm gonna get real speculative here. Okay, I'm gonna talk about antibody specificity and antigenic drift. And so I want to go back to this slide. I set this up earlier that all of us have different antibody specificities to flu depending on the specific type of influenza strain that we saw as a kid. I want to show you some data on this. So this is a paper from Susie Linderman, who was a great grad student in my lab and now is a postdoc with Rafi Ahmed. And okay, so this is some flu language here that's not important. Just the y-axis is percentage of serum samples that have a specific type of antibody. Just leave it there. And then on the x-axis are a year of birth of donors of these serum samples. Okay, so type of antibody response, year of birth. And what you can see is that people born prior to 1985, in particularly these people born in the 1970s, about half of these people had this specific type of antibody response that was recalled by the 2009 pandemic H1N1 virus. And the reason I want to show you this is look what happened for those born after 1985. None of these people had this antibody response. I think this is super interesting because it's a cliff, right? If you were born prior to the 80s, you had this type of antibody response that involved an epitope involving this residue K166. But if you're born after 85, you didn't have that specificity. Well, what was going on here is a structure of hemagglutinin. So in 1983, this is what hemagglutinin looked like, and here's the 2009 pandemic. In red are residues that differ between the 1983 virus and the 2009 virus. So when individuals were primed with 1983 and then boosted with 2009, there was this focused antibody response against this one epitope. And why was that the case? Well, look at that epitope. It's completely conserved between these two viruses. Old school immunology. Tommy Francis would love this, right? You get primed in the 80s, and now you get boosted in 2009. You get a focusing of your antibody response against the conserved antigenic sites. Okay, and as I was kind of muddling about a few slides ago, if we would just take antibodies, collected in 2008, we wouldn't really see these antibodies. They weren't really prevalent until this boost, okay? Because when you get a boost with two different things, you enrich for these what were rare antibodies and now become dominant antibodies. Well, what happened in 85? What the heck? What the people that were born in 85, why didn't they have this antibody response? Well, this is what a 1985 virus looked like. Looked very similar to an 83 virus. Shown in red are residues that differ between the 2009 virus and the 83 and the 85 virus. But guess what? It had this lightning bolt here. This lightning bolt's a glycosylation site. So the 85 virus had this glycosylation site, which essentially shielded this antigenic site. And so people born after 85, when they got infected with 2009, they didn't have this antibody response at all, because when they were kids, they never had priming of B cells against this site, because that site was covered in the form of glycosylation. I think this is remarkable. So there we have it. Going back to this. So what happened? So everyone has different immune, imprints, and now you get a focusing on these epitopes that are conserved. What happened to the 2009 virus? We still have this virus. It still circulates. Well, several years after circulation, this is what happened. Everyone see that graphic? The virus acquired a substitution in a green antigenic site. And when that happened, middle-aged individuals became more susceptible to the virus. And we published this paper in PNAS a few years after, I don't know, in 2014 or something. So here's residue 166. This is a key residue involved with this antigenic change that I'm telling you about. In 2009, it was a lysine. And you can see during the 13-14 season, there was a change, a K to Q change at residue 166. Concurrent with this antigenic change, middle-aged individuals became preferentially infected with the virus. There was more disease in that age group. Why? Because they had antibodies that could react, that were recalled to the 2009 strain and helped them recover from 2009, but could not fight off this drifted form. And so Patrick Wilson has made monoclonal antibodies. Here is a monoclonal antibody from a middle-aged individual. It can react to the wild type strain, the 2009 virus strain, but couldn't react to that drifted strain. When we passively transfer these antibodies into mice, you can see that this antibody can protect against wild type infection, does not protect against this drifted infection. Here's a control antibody that recognizes outside of that site. And we did correlates of protection type of study. So in this case, this is a collaboration with University of Michigan, where we had 382 humans that were bled prior to this flu season. 20 of these individuals were naturally infected with H1N1. The question is, well, what kind of antibodies did these 20 people have before they were infected? We stratified these individuals into three groups. Those that didn't have antibodies to that original 2009 pandemic virus in the drifted form, that's these guys. They didn't have antibodies to either strain. People that had antibodies against both strains. And then here's the kicker group, people that had antibodies against that 2009 strain, but they failed to react to that drifted strain. There are people I just told you about, like Yang Li. Who got infected? Who are those 20 people? Whoa, right? So if you don't have antibodies during the 13-14 season, there's infections in that group. If you have antibodies against the 2009, that drifted strain, well, you're protected. But here's the important group. We had 41 individuals that had antibodies against that wild type strain, but couldn't recognize that drifted strain, and they were just as susceptible to those that didn't have antibodies at all. Okay, so we've been working with Jesse Bloom, and Jesse Bloom is an amazing scientist. If I was going to pick someone to have a beer with as a living scientist, maybe it would be Jesse Bloom. I really like Jesse. I like his work. He came up with this deep mutational scanning approach to map the specificity of antibodies against influenza strains. And this really, we were on this paper because we helped him with some antibodies, but this is really his work and his grad student Michael's work. And so what Jesse did was he has a deep he makes these virus libraries that each have a single substitution in hemagglutinin, and he can then passage these libraries in the presence of antibodies or just mock no antibodies, and then he sequences what comes out. And the viruses that come out here at the end in the presence of antibodies, well, those are viruses that have substitutions in key antibody binding sites. And we published this paper with Jesse, I guess towards the end of 2019, where Jesse used this technique to map the specificity of antibodies in polyclonal serum. And what you can see here is in line with what I've told you about some of our prior studies, that different aged individuals select for or possess antibodies that react to hemagglutinin in different residues. Okay, so shown here in these stacks of letters. So for example, in this individual, upon passaging the virus in the presence of his or her serum, viruses had this substitutions at this particular residue. I don't really see what residue this is enriched, and this residue enriched, but this 53-year-old didn't have very much selection at all. See how that works? I'm doing a terrible job describing that. But nonetheless, now when you could do neutralization assays, you could use reverse genetics and now make homogeneous stocks of these viruses and do neutralization assays. You can see this 21-year-old man cannot recognize this light blue virus at all, but can recognize this green virus. But this 64-year-old cannot recognize this green virus, but can recognize a blue virus. So again, consistent with what I've showed you, this technique can be a very powerful technique to probe the specificities of antibody types that are prevalent in polyclonal serum samples. And Jesse, man, he has been doing some good stuff with COVID-19. And I think many of you are probably familiar with this paper that was put on medRxiv maybe a week or two ago. Used a similar technique, in this case, a yeast library approach, to map the specificity of antibodies in polyclonal antibodies in different individuals. And here you can see that when subject A, these libraries were selected with serum from subject A, there was an enrichment, for example, in substitutions at residue 456. But when the same experiments were done with serum from subject C, you didn't have that much enrichment for 56. This is SARS-CoV-2 now, the spike protein, the RBD domain. Instead, you have residues selection at 484. So there's a lot of variation, okay? And similar to the flu studies, Jesse went on to show using neutralization assays that some of these substitutions had great effects. And that there's a lot of variation between individuals. And we're certainly concerned about some of these because residue 484, for example, is a substitution that is in some viral lineages that are emerging now. So I'm going to end with speculation. Why is there so much person-to-person variation in the specificity? It could be due to polymorphisms in the heavy and light chains. Okay, in the case of flu, we know, for example, that hemagglutinin stalk antibodies, that there are certain polymorphisms in VH169 that promote effective group 1 HA stalk antibody responses. So similar stuff, I'm sure, is going on to some degree here with SARS-CoV-2. It could have to do with antigen dose, disease severity, how long was the antigen around. These are things that we're interested in looking at. But of course, it could have to do with pre-existing immunity. And so Trevor Bedford and others have been studying, it's well documented, I guess, now that there is clear adaptive evolution in the RBD domains of seasonal coronaviruses. And so at least that idea of, Yang Li might have been exposed to a different coronavirus compared to Tony Fauci, at least in childhood, at least that idea is possible because it's clear that these viruses have undergone antigenic change, albeit at a much slower level compared to influenza viruses. And when you look at the structures, I mean, look, OC43 is different than SARS-CoV-2 for sure. But if you do an overlay, it's not like these viruses, again, are just from like different planets, right? They do share some homology. Granted, most of the homology is in S2, but I wonder if there is some homology that we still that is underappreciated in some of the S1 domains. This part's to be continued, right? This is exactly what we're working. Why is there so much person-to-person variation? How does exposure history shape the antibody specificity to SARS-CoV-2? And is the virus evolving to avoid antibody pressure in a subset of individuals? Or is it simply evolving to optimize receptor binding avidity? And that subsequently is affecting antibody binding, okay? We don't know. It's hard to figure out these things. But the virus could simply becoming more, maybe it's just becoming more transmissible, right? And some of these residues have been shown to affect ACE2 binding. Maybe it's increasing virus attachment to evade polyclonal antibody responses. And maybe these residues, it's sort of a side effect that they also affect antibody binding, because just like flu, many of these residues involved with virus attachment are also involved in antibody footprints. And that's because most neutralizing antibodies actually sterically inhibit antibody binding. So there's a lot to learn here, but I think doing these kind of scanning approaches that Jesse and others are doing to try to and doing large scale sequence analysis to try to figure out which viruses are out there, and trying to understand the immune landscape at a population level, looking at within individuals, between individuals, looking at trends of different age groups and whatnot can lead into us being able to predict better about how some of these substitutions are going to affect the ongoing pandemic. Finally, we're very interested in looking at infection versus vaccination. There's reason to believe that antibody specificity might be different there. Okay, I realize I'm running out of time, and I want to make sure that I have time for questions, but I do want to talk just a minute or two about racial and ethnicity differences in SARS-CoV-2 exposure. Today, my talk has been geared more towards basic science questions of prior exposure, but my lab, during the early days of the pandemic, we basically just turned into a factory to try to complete clinical studies. We enrolled 2,000 healthcare workers to measure seroprevalence and exposures in our hospitals. We completed these large seroprevalence studies, which I'll show you a bit today, in pregnant women to try to understand the scope of the pandemic in Philadelphia. And so today I want to just show you a few data slides on how these seroprevalence studies have shed light on some of these really terrible differences in SARS-CoV-2 exposures as it relates to race and ethnicity differences. So we started with this question: what's the overall seroprevalence in Philadelphia? And it's a tough question to answer in the middle of a pandemic because it's hard to get blood in an unbiased way. And there are several groups of people that continue to seek healthcare, even in the middle of a pandemic. One of those groups are pregnant women when it's time to deliver their baby. Okay, regardless of income, regardless of race, most women, when it's time to deliver a baby, go to the hospital. They don't just decide to deliver via Zoom, right? And so we thought this was a way to unbiasedly measure antibodies in a wide population. And so you can see we collected, this was during the first wave of the pandemic, over 1,000 samples for pregnant women. We basically captured all 50% of births in Philadelphia among two of our academic hospitals associated with the University of Pennsylvania. And so here, after the first wave, we found about 6% of women were seropositive. You can see IgG and IgM levels. And this now, so we continue to do this. We do this every week, and now we're up to something like 12 or 15%. Just real quick, so this paper just got will come out, I think, in a week or two. There's this narrative that maternal antibodies are not effectively transferred. That narrative's not right. I don't understand some of those studies. What we found is the transfer ratio. So if you look at antibodies in the infant compared to antibodies in the mother, you can see this is a nice straight line. And then if you look at transfer ratios, the transfer ratios are pretty high. This is known in viral immunology. If you're recently infected or have an ongoing infection, your transfer ratios are not that high. And that's been reported recently in with SARS-CoV-2. And that has been sort of reported as this is something unusual to SARS-CoV-2. No, early, if you're actively infected, you have an IgM response that doesn't get across the placenta. But you can see that as time goes by post-infection, the transfer ratios become much higher. Again, this is something that is not new to SARS-CoV-2, but it should be reassuring because, we'll have to start thinking of, well, everyone is starting to think already, should we vaccinate pregnant women? How much protection might it afford to their offspring? The reason I want to talk about the seroprevalence study today, though, is not just to look at seroprevalence over time. I told you that this has reached something like 12 to 15%, but this slide right here, okay? again, regardless of your race or ethnicity, most women are delivering babies in a hospital. And when we look at seroprevalence, this is during the first wave of the pandemic, but we're seeing similar things in Philadelphia during our second wave, that Black women and Latino women have something like a 5-fold higher increase in seropositivity compared to white women and Asian women. That is remarkable. When we know that COVID-19 is disproportionately affecting Black and Latina and Latino communities, is that because of differences in comorbidity? No, maybe comorbidities and stuff like that. I'm sure it's complicated, but without a doubt, it's at least impacted in exposure. Okay, if populations have 5-fold higher levels of exposure, then of course that's going to lead to more disease. And why are individuals of different race and ethnicity exposed more? Well, it's deep rooted in systemic racism. Okay, and these are the things that we're trying to figure out now. I'm going to leave it at that, but this is something that is, when I saw these data, I was horrified, but individuals that actually study health disparities in medicine, this is all too familiar, okay? And so we need to, we really need to think about ways to, minimize this not only for SARS-CoV-2, but just, all different aspects of medicine. Okay. I think I've left 10 minutes for questions. So early in the pandemic, I told you we became a factory, right? And here's Maddie, a technician in my lab, hugging a bunch of pipette tips. We received funding from a healthcare worker study from the 76ers and the Eagles, and Joel Embiid, who's a star center on the Philadelphia 76ers. I didn't talk about that healthcare worker study, although it's a fantastic study, maybe another time. And I'd like to just thank folks in my lab, as well as folks all across Penn. It's just been, a real honor to work beside these individuals during this pandemic. So with that, I think there's 5 minutes for questions. Thank you for having me.
Thank you, Scott. That was a great talk. I almost feel like taking an immunology class right now. So let me start the Q&A with a very simple, maybe a naive question here. So we heard a lot about antibodies, some of which could be neutralizing or not. So referring to what you showed right at the beginning of the talk, the pre-existing antibodies from childhood infections. So are these antibodies from childhood infections, are they neutralizing as well? Or is their sole function to boost the antibody production in the current infection?
Yeah, so in childhood, what we think is that you have a somewhat broad antibody response induced. That includes B cells that will go on to differentiate to produce antibodies against both neutralizing sites and non-neutralizing sites. The problem is that during a pandemic, the reason these viruses cause pandemics is that their neutralizing sites are so different. And so during a pandemic, you have a situation where the B cells that can respond, that have BCRs, that have some level of homology to be able to recognize that pandemic virus, that they tend to recognize the non-neutralizing sites. So we think childhood infections probably establish memory responses, long-lived memory responses against neutralizing sites and non-neutralizing sites. But when we're faced with a pandemic virus, the only homology is in the non-neutralizing sites, and those tend to get expanded. It's also a problem with flu, because as you age, then the viruses become more antigenically dissimilar. And we think that a major reason that old folks are more affected with flu is because they get infected with viruses that differ in neutralizing sites to whatever they were exposed to as kids.
So audience, be sure and either ask your question in the Q&A box or raise your hand and we will call on you. Scott, if you don't have a hard stop, we can stay for a couple of minutes after. So yeah, I've got a lot of questions here.
Brian.
I want to jump right in and get your take on, okay, so this is a bit of a complicated question. So there's a fair bit of evidence emerging that SARS-CoV-2 can inhibit formation of germinal centers, particularly in people that get severe infection. Actually, a lot of autopsies show that the patients had no germinal center formation in their lymph nodes. And that, of course, is the nexus of antibody hypermutation, meaning that a lot of the antibodies have fewer mutations relative to germline than for your average pathogen. So, and I think this is emerging in the antibodies that have been sequenced. They don't really have a ton of mutations relative to what one had before. So I'm wondering how you think about that in this disease.
Well, Loren, I don't know if all of what you just said is known yet. My understanding is that there is actually a fair level of somatic hypermutation in a lot of these antibodies. Patrick Wilson has a preprint online that shows that many of these, he's made monoclonal antibodies from COVID-19 survivors, and many of these antibodies have high levels of somatic hypermutation. There was also a paper that was just published yesterday in Nature from folks at Rockefeller, and haven't had a chance to get into that in too much detail. But in that study, I think it's looking over the course of 6 months post-infection. What they see is an evolving antibody response over time. And something I think is really interesting is, evidence of persistent nucleic acid, at least in the gut. So, I think it's a little bit too early to say any of these things definitively. But I think taken together, it seems that at least a fraction of the antibody response upon SARS-CoV-2 infection is from the memory component. And that, over a course of an infection and, persistent antigen over time, that there's probably germinal centers that, that get established and you have additional substitutions occur.
Yeah, we had, but Paul Binosh was in our seminar series, and he showed us that amazing data. I mean, my read on his opinion on that was that the subsequent mutation was likely being initiated from the gut, and then coming back to plasma IgGs. But yeah, you're right. This is definitely not nailed down yet.
I tell you, a lot now that the vaccine is becoming more readily available, not as available as some of us would like, as I sit here in my basement. But I think we're going to be able to do some really nice studies with studying the vaccine. Vaccination might be different than infection, but having pre and post PBMCs and being able to track specific clones, for example, we're going to be able to do some elegant studies with pre and post vaccination samples to get at these questions a little bit better.
All right, we've got some audience questions.
Ryan, please unmute yourself.
Thanks so much. What a great talk. I have a question that you probably can't answer definitely, but what is your feeling? Is this going to be like the flu? We're going to have to get a COVID-2 vaccine every year, or what? How likely is that scenario in your opinion?
Yeah, I don't know. It seems clear that these viruses do change over time. If you look at other coronaviruses that are endemic, there's evidence of SARS-CoV-2 changing already. I can see a scenario where we have to update the vaccine, every few years. My hope is that we can get this thing under control. If we have, enough immunity through vaccination, and we can keep virus levels lower, that there wouldn't be such opportunity for the virus to explore the genetic space required to have some of these antigenic changes. And it's, if you take, you take one shot on goal, I don't play hockey, I don't know anything about hockey, but you can put me in a hockey rink with a professional hockey player and you give me one shot. Well, guess what? It's not going to go in, right? But give me 400,000 shots and you know, I'll probably slip one in somehow. And it's the same thing with viruses. You know, you have hundreds of thousands of infections in the US every day. Well, that's a lot of opportunity for some of these, you know, some of these random variants to emerge. And so I think how it might play out is that we do see antigenic change somewhat soon, since the virus is exploring so much space right now. But my hope is that as things die down over the next year, that virus levels can be kept at such a low level that it becomes more rare for some of these variants to emerge.
Next, we've got Rafael. Rafael, okay, please unmute yourself.
Hi, Scott. Nice talk. I just wanted to tickle your mind a little bit with glycosylations. Of course, the SARS-CoV-2 protein is highly glycosylated. I haven't seen in detail if any of the changes have arisen with the variants might alter the glycosylation pattern of the spike. But what is your take on the potential role of glycosylations in SARS-CoV-2 antigenicity?
Yeah, that's a good question. I mean, glycosylation, you know, sugars are big, right? And these glycosylations can have much bigger effects compared to single residue changes. I mean, of course, single residue changes, if they are right in an antibody footprint, can completely abrogate antibody binding. But glycans, you know, can have larger effects more distant away from where that glycosylation site actually is. So in the case of flu, we have examples of single residue changes that do not affect glycosylation that have big effects. And we also have cases, for example, the H3N2 viruses, these contemporary H3N2 viruses that circulate now. One reason the vaccine is so ineffective, our current H3N2 vaccines are so ineffective, is because of a mismatch in a key glycosylation site. So this will be something important to study further. Now, one thing about glycosylation, I'll tell you, is that even though glycans can have profound antigenic effects, they can also affect a viral protein function quite a lot. And so, you know, in case of flu, it kind of has to balance that as the virus acquires the glycans, it has to, that they usually have effects on receptor binding avidity. So there's a balance there.
Excellent. Thank you, Rafael. Scott, I've got another one. I'll make this one much simpler. So you showed the really cool data from Trevor about the natural history of OC43 evolution. Did they map where those mutations are occurring and trying to assign, you know, just ballpark putative function? Do you think it's mostly antigenic escape or, you know, infectivity, et cetera, et cetera?
Yeah, so I'm still trying to learn about this as well. So I think previous studies have identified a few key epitopes in OC43 viruses over the course of, you know, the last 40 years or so. So I'm not an aficionado yet in this, but my understanding is that there is, you know, that there's evidence of antigenic evolution over, you know, the last, you know, several decades, that antigenic change occurs slower. And, you know, I think there is, again, this is something that I'm still reading papers on actively and trying to inform myself. But my understanding is that there are mapped, you know, loops and whatnot that are important in mediating some of the antigenic change in some of these older coronavirus. Now, you know, if you compare OC43 RBD domain compared to SARS-CoV-2, some of the loops just don't even exist, and that's because you know, just ACE2 binding. You know, OC43 doesn't even engage ACE2, is my understanding. So we are trying to understand that now. Are there possible structural areas of structural homology between these two very different RBD domains, and has that changed over time with OC43?
Excellent. Okay, Sarada's got another one now.
Yes. So Scott, switching gears a little, since we are trying to learn lessons from the influenza virus, you probably have a postdoctoral paper where you see that the influenza virus, especially in immune host, it starts to acquire mutations that make binding more efficient. And now some of the SARS mutation that we see are also are binding to ACE2 more efficiently. So, and some of the viruses in the influenza that you've seen are also probably efficient in evading immune response. So do you think the new viruses that we see in SARS-CoV-2 that are binding to ACE2 more efficient are also probably could be efficient in immune response, evading immune response, pardon me?
Yeah, so it's a good question. So when I was a postdoc, what we found in the case of flu is that flu viruses, if they're confronted with polyclonal antibody responses, antibodies against many different epitopes, the virus polymerase error rate's not high enough where it could just acquire 5 substitutions simultaneously. But the error rate it requires 1 HA substitution on average. So when confronted with polyclonal antibodies, flu viruses just can't evade everything, and so it does the next best thing. And the next best thing flu viruses can do is to acquire single substitutions that allow viruses to attach to cells with a higher avidity. If a virus binds with a higher avidity, in essence, that lowers the neutralization of antibodies of all different specificities, at least those antibodies that block receptor binding. Because, those kind of antibodies, it's a balance between antibody binding, actually blocking, and then the virus actually binding to the cell. In those cases, we've learned that influenza viruses with higher avidity are favored in populations where there's more immunity. And then interestingly, we found that those same viruses, then, if they're thrown into a naive population, that they are less fit, and that viruses with lower avidity are actually favored, lower receptor binding avidity are favored in naive populations. And the thought is that if there's no immune pressure to deal with, a virus would rather bind sort of loosey-goosey, because viruses that bind too tightly to cells, they're not effectively transmitted. They're not effectively released at the end of the day. So it's nice to, it's interesting to try to speculate that some of these residues that we see emerging now might be in response to polyclonal immune pressure in the population, that viruses are binding tighter to try to avoid antibodies of all different types of specificity. And that is in agreement with population worldwide immunity increasing, but we just don't know. It's possible that these substitutions are just emerging to allow the virus to become more transmissible, to optimize ACE2 binding, and so it can replicate in the upper airways. And as a byproduct of that, some of those ACE2 modulating changes are also going to concurrently affect antibody binding. And so the ones that get selected might be the ones that do both. What I presented today is this idea that some of us walk around with very focused antibody responses. So you can kind of think of viruses as having at least two ways to evolve. If there's polyclonal antibodies against many different sites, viruses tend to just acquire substitutions that allow it to grab a hold of the receptor with higher affinity. But if some people are walking around with these super skewed responses, as Jesse Bloom's work, I think, has suggested, at least some people are very focused on particular antigenic sites. If that's the case, the viruses will often just acquire single substitutions that abrogate antibody binding. So we don't really know yet, but it is interesting. And one thing is for sure, that the residues involved with both antibody binding and receptor binding to ACE2, they're the same residues. So it's hard to disentangle these things. But those are the kind of studies that we want to do to try to figure out how the virus evolves when confronted with different antibody pressure. And I think what's really important now, and something that we are getting geared up to do here at Penn, is to really try to understand the clonality of antibodies in the population. Is it 10% of us walk around with antibodies against 484, or is it 5%? Is there an age group that has particular types of antibodies? Those are the kind of questions we want to know.
Interesting. So just one quick follow-up note. We keep talking about point mutations, but the 3 amino acid deletion seems to be a hallmark of all the variants that you're seeing so far, the 3 amino acid deletion in ORF1AB. Is there any clue what's going on with that?
Yeah, I don't know. I mean, deletions can definitely have antigenic effects as well, right? So, case of, I know much more about flu than I do coronaviruses. In the case of flu, there are definitely documented antigenic changes where we see insertions or deletions. So my understanding is that that the deletion that you're talking about is one that we have to keep our eye on.
True.
Yeah, Scott, this has been amazing. Can you maybe just leave us with a final thought about what do you think about the current variants that are being billed as, you know, on their way to antigenic escape and potentially even escaping from polyclonal serum? Should we be worried or panicked yet? And how do you see this playing out?
Yeah, so everyone should get vaccinated. I think we have to work with our governments around the world to increase vaccination. It seems insane to me. You look at the U.S. and we're not sure how to distribute a vaccine, as if this problem just occurred in the last few weeks. I mean, in my mind, we should have been shipping little syringes filled with saline in the spring just to give a practice run. So now that we have a vaccine, the most important thing to do is to figure out how to distribute it, get it into people's arms, and how to ramp up production and make sure that this never happens again, right? That we are so ill prepared of scaling up a vaccine and distributing it. It's a crime. All the science that went into making this vaccine, now you can't get it to people. So everyone should get vaccinated. Should we worry? Of course, I worry about things, but if we are all vaccinated, again, we are going to, if you think about that ice hockey analogy, we're going to limit the space that the virus has in randomly being able to acquire substitutions. And so we all have to join together and get vaccinated, try to get these virus levels down to levels. And look, if let's worry about antigenic drift later. I mean, at the end of the day, most of us probably have somewhat of a polyspecific response. And even in Jesse's studies, for example, if you have a reduction of neutralization by tenfold, it doesn't go to zero, right? There are still some neutralizing antibodies, even in those individuals that tend to have focused responses. And, there's other things like T cells, right, that might be involved with protection. So everyone should go get vaccinated. Let's worry about getting the current vaccine to everyone else that hasn't had a chance to get it. And let's monitor these things. The great thing about mRNA vaccines is you press a button and now you can update the vaccine, right? It's synthetic. So if we do need to update these vaccines, it shouldn't be a real hard thing to do.
Yeah. Well, again, thank you so much for your time and the work that you've done.
All right. Well, thank you for having me. Take care, everyone.
Bye-bye.